In this next video, we are going to be talking about some accessory external structures that you may find on bacteria. So an accessory structure means that the bacteria does not need to have this structure in order to survive. So some may have these structures, some bacteria may not, okay? Because they're external structures, that means we find them outside of the cell, okay? So far we've talked about the plasma membrane, which is the border of the cell. Then outside of that is the cell wall. So now we're going to be talking about structures that might be outside of that, okay? So a lot of pathogens have what's called the glycocalyx. So notice there's the word glyco in there. Remember, glyco means carbohydrate or sugar. So the glycocalyx is a sugary layer that is sometimes outside of that cell wall. Now, when this sugary coat is really firmly attached to the cell surface, we refer to it as a capsule, okay? So in this class, that's all we're going to talk about in terms of glycocalyx is when it's firmly attached. So you're normally just going to hear me refer to this structure as the capsule. Okay, but please know it is a sugary coat. So glycocalyx and capsule in this case can be used interchangeably. Remembering that it's a sugary layer is important, okay? And so because it is a sugary layer, it can, under dire circumstances, be used as a form or a source of nutrition, okay? But it can also help organisms form a biofilm. We talked about biofilms during the Intro to Micro series. And remember, biofilms are a community of organisms. Different organisms can get together and hang out with one another, forming a little community. And that's what you see on this picture right over here, this bottom picture. Trying to get my mouse to cooperate. There we go. So if you look at the far left of this little like microscope slide is what it looks like to me, you'll notice there's really just one type of bacteria over there. If this bacteria has a capsule, that sugary layer, it's going to be sticky. So it can adhere to this surface. Notice it's starting to form a little hydrogel around it. Okay? It can recruit other organisms to come and be a part of this community. And biofilms tend to grow up and out. Okay, so notice how it's getting bigger and bigger over time. And this is a real issue in hospital settings. The science term for that is a nosocomial setting. So in healthcare settings or nosocomial settings, biofilms can be problematic, especially in people that might have a catheter or a heart valve, because as these biofilms get bigger and bigger and they're sticky, they can clog a catheter, they can clog a heart valve or other, similar things. And so, right, problematic. They are also problematic because of this like hydrogel that surrounds them, kind of this yellowy layer that you see on the outside of this biofilm. That hydrogel is pretty thick, okay? So let's say we have an infection and the pathogen that has gotten into our body and made us sick is part of a biofilm. Well, when we take antibiotics, it's going to be really hard for that antibiotics to penetrate through that hydrogel. Okay, that hydrogel is kind of protecting those bacteria. So it's going to be harder to eliminate those bacteria compared to other bacteria that are not part of a biofilm. Okay. Now, we also say the capsule is a virulence factor. Okay, so here's that word again. So remember, virulence factors are kind of weapons that the bacteria can use. So far, we've kind of talked about structural virulence factors, that LPS, that endotoxin, that's part of the gram-negative cell wall, for instance. But the reason the capsule can also serve as a virulence factor is because it is sticky. It's a sugary layer. Well, it turns out when we get infected by a bacterial pathogen, or any pathogen really, our immune system is going to respond. Part of our immune system involves phagocytic cells. And these phagocytic cells do a process called phagocytosis. And during phagocytosis, these immune cells are hunting down the bacteria. They're going to grab hold of the bacteria. They're going to ingest them and digest them, right? They're kind of chopping the pathogen to bits, getting rid of it so we can start to heal. So that's the normal process, okay? But when the bacteria has a sticky capsule, it can be really hard for that immune cell to grab hold of and capture that bacteria, okay? So we can say the capsule helps the pathogen escape or evade phagocytosis, okay? And that's bad for us, but very good for the bacteria because the bacteria don't want to be chopped up and eliminated, okay? So the capsule has offers a lot of advantages to pathogens. And so you see a lot of pathogenic bacteria do have capsules. And in some instances, if there's a strain of that bacteria that doesn't have a capsule, it actually becomes non-pathogenic. So the capsule is pretty important for a lot of pathogens. All right, my favorite external structure, the flagella. So a flagella is a long whip-like structure that rotates much like a boat propeller, okay? And so it helps propel the bacteria through the environment. And so as we'll talk more about in a minute, some bacteria don't have flagella. Remember, this is an accessory structure, but there are different arrangements of flagella that a bacteria can have. But whether it has a single flagella or a ton of flagella, they all move, okay, kind of in one direction, again, like a boat propeller. And that's how they move in a straight line. However, and we call that process running, okay? And so whenever the bacteria wants to change directions, though, it has to start trying to move the flagella in the opposite direction. So think of a boat that is moving in a straight line through the water, right? Let's say the propeller is moving clockwise. If you suddenly were able to make the propeller start going counterclockwise, right, there'd be some resistance because it'd be going really quick one way and it would be kind of stuttering through the water trying to go the opposite direction really quickly, right? So it would kind of stutter. And so that happens to the bacteria as well, but we don't call it stuttering, we call it tumbling. I kind of think of it as the bacteria looks like it's spazzing out, trying to get all of its flagella to start moving the same direction at the same rate. And it eventually will, and we then say it's running again because it's going again in a straight line. So this is actually how bacteria can try to find foods or just be attracted to some sort of stimulant. So as you see in this top picture, the bacteria is running in one direction. For whatever reason, when it needs to change direction, the flagella try to move the opposite way, so it kind of spazzes out. It tumbles, and then eventually it gets it together, and it will start running again. So run and tumble are associated with flagella and their motion. So when we talk about flagellar arrangement, this is important because it's how we can classify bacteria. So some bacteria do not have flagella at all, and then others might have multiple flagella in multiple locations. So let's first look at what we call a bacteria that has one single flagella. The term for that is monotrichous. So mono means one. Trichous, I believe, is Latin for hair because flagella look kind of like a hair. And so it has one hair. Okay, so one flagella, monotrichous. Amphi means both. So this means there's a flagella at both ends. Okay, so anytime you see trichous, you need to think of the word flagella. Amphi means both. Okay, so there's a flagella at both ends with an amphitrichous organism. If there's a tuft or a cluster or a group of flagella only at one end of the bacteria, we refer to that as lophotrichous. Okay, so lophotrichous means there's a tuft of flagella at one end of the cell. And then the other arrangement is peritrichous. No clever way for what peri means, but how I think of it is a peritrichous organism looks really hairy, right? So hairy so peritrichus looks hairy. So that's how I remember it personally. So then if the bacteria doesn't have any flagella at all, it's atrichous. Because remember, when you have a at the beginning as the prefix, that means without. So it literally means without flagella. Okay? So I love flagella. It's one of my favorite structures. So make sure you're very familiar with this information. Hint, hint, wink, wink. All right, and the final two structures, again, accessory structures, are Fimbriae and Pili. So Fimbriae are these little short hair-like projections that will cover the entire organism. So if you look at this picture, I noted some of the Fimbriae. Looks kind of like a shadow in this picture, but if you look close, you can see there are these tiny hair-like projections, but they do completely cover the bacteria. Okay, if you look at where my mouse is, you can see there's Fimbriae all around the bacterial cell. The purpose of a Fimbriae is to help with attachment. Okay, they kind of stick on to surfaces. So they can also help facilitate the formation of biofilms as well. And Fimbriae are only found on prokaryotic organisms. Okay, so it's unique to prokaryotes and it helps with attachment or adherence. A pilus, on the other hand, is a long structure. It kind of looks like flagella, but to me it's more of like a tube-like structure that some cells use to literally attach to and rein in a neighboring cell. Okay, so one of these cells has essentially grabbed hold of the other and it's going to draw it close to it. Because what we're going to talk about in a future video is a process called conjugation, which is one way that a bacteria can acquire DNA from a different bacteria. Okay, and so in order to do that, the bacteria need to be close to one another. So a pilus can be used, I think of it as like a fishing line. Right? When you go fishing, you cast out your hook. So in this case, it will extend out a pilus or pili if there's multiple. One cell extends it out. It'll grab hold of another bacteria, so like a fish in my analogy here. And then what do you do if you do grab a fish? You then bring it into you. And that's what's going to happen is the cell that released out the pilus, once it grabs hold of a neighboring bacteria, it brings it in so that they're neighbors. And then in more detail, we'll cover later, they can share DNA. So it's pretty cool. So the pilus, again, helps attach to a neighboring cell and bring it close to one another. All right, y'all, that is the end of our discussion on external accessory structures that you may or may not find on bacteria. Let me know if you have any questions.